In 1928, Alexander Fleming made a groundbreaking discovery that would change the course of medicine: penicillin. The event occurred when he returned to his laboratory at St. Mary's Hospital in London after a vacation and noticed something unusual on a petri dish containing Staphylococcus aureus bacteria that had been left uncovered. A mold, later identified as Penicillium notatum, was growing on the dish, and Fleming observed that the bacteria surrounding the mold were being destroyed while those farther away remained unaffected. Fleming conducted further experiments and isolated the mold, determining that it produced a substance capable of killing bacteria, which he named penicillin. He published his findings in 1929, noting its antibacterial properties against various gram-positive pathogens. However, despite his significant discovery, the medical community largely dismissed penicillin as a laboratory curiosity at that time. Fleming struggled to purify penicillin in sufficient quantities for therapeutic use, and his attempts to enlist help from chemists were largely unsuccessful. It wasn't until nearly a decade later, in the late 1930s, that Fleming's work caught the attention of scientists Howard Florey and Ernst, who recognized its potential for clinical application. In 1939, a team at the University of Oxford, led by Howard Florey, began serious research on penicillin, focusing on its extraction and purification. They conducted animal testing to confirm its efficacy and safety. The first human trial occurred in February 1941 with an Oxford policeman suffering from severe infections. Unfortunately, the supply of penicillin was insufficient for complete treatment, leading to the patient's death. The outbreak of World War II intensified the need for penicillin, prompting collaboration between British scientists and American pharmaceutical companies to scale up production. Techniques were developed to grow the mold in deep fermentation tanks, significantly increasing yields. By 1943, mass production was underway, providing essential treatment for wounded soldiers. Penicillin's introduction revolutionized medicine by drastically reducing mortality rates from bacterial infections. For instance, the death rate from bacterial pneumonia fell from 18% in World War I to less than 1% in World War II due to effective penicillin treatments. The antibiotic became widely available post-war, leading to further developments in antibiotic therapies and the discovery of other antibiotics such as streptomycin and tetracycline. Fleming, Florey, and Ernst were awarded the Nobel Prize in Physiology or Medicine in 1945 for their contributions to the discovery and development of penicillin. Fleming's initial observation not only marked a turning point in infectious disease treatment, but also initiated an era of antibiotic research that continues today. The legacy of penicillin is profound. It not only saved countless lives, but also established the foundation for modern pharmacology and the ongoing battle against antibiotic resistance.